Thanks, Lee, and thank you very much for the invitation to give a seminar here. Normally when I sort of flit through Brisbane, which is on a semi-regular basis, I usually try and keep a very low profile, but it made good sense to sort of come and learn a little bit about what you're doing. We had Jack Christopher from here give a seminar in Lancaster only a couple of months ago, and it seemed reasonable that I should reciprocate. It's great to see so many familiar faces in the audience today. It's very much like coming home. I'm afraid I'm going to start with an apology. The title, Root System Distribution and Root Shoot Signalling, is perhaps not strictly accurate. What I'm mainly going to concentrate on is soil moisture distribution and root shoot signalling. But based on the premise that roots generally go where there is soil moisture, I think we can extrapolate. And certainly I'll be at pains to point out those times where we have got some root information. So this is Lancaster here. This is the campus. And you can see that it's in a sort of fairly green pastoral area. Over on the sort of coast there, you have the premier seaside resort of Morecambe. And on a good day, if you time it absolutely perfectly, you can go there and have a swim. If you time it wrong, the sea will be about 10 kilometres out from the beach. It's a tidal environment, and it's a site of special scientific interest for migratory birds. And you can see in the background the Lake District National Park with snow on the hills, a very nice part of the world to be. Based on this, if I were to tell you that I was interested in the impacts of abiotic stress on plants, I think you would probably expect to see an image such as this. This is not Lancaster University, this is Myerscough College, which is just south of Lancaster, and it's where we do our field trials. Now, I'm most interested in the impacts of soil water deficit on plant growth and development. And so to do field experiments in this part of the world, we have to take special precautions. So this is some work we did on wheat a few years ago, and you can see that to actually prevent the weather spoiling our experimental design, we actually need to put these rain-out shelters up to ensure that we impose the treatments that we're interested in imposing. However, I don't want you to think that crops grown in the UK are completely without water stress. I'll show some data later when it comes to the wheat section of the talk to show that if you irrigate in the UK, particularly on the lighter soils, you can improve yields by about 1 to 3 tonnes per hectare, which I suspect by an Australian standard is actually quite a significant improvement in yield. So if you are going to irrigate to improve yield, it begs the question, how do you do this most efficiently? And certainly, if you were to go out and survey farmers, I suspect you would find a huge variation in how farmers actually are applying irrigation, when they are applying it. And indeed, this is what Elias Ferreras, who's in Spain, in Cordoba, has actually done. He's gone to this irrigation district and interviewed the farmers that were growing sunflower and actually tried to understand when they are irrigating. And if we look at this graph here, we can see we've got rainfall in the sort of blue bars, and then each one of these individual lines actually indicates the percentage of the total annual irrigation that the farmers are applying. Now you can see in some cases, farmers are applying all their irrigation water at the beginning of the season. In some cases, all their irrigation water is applied at the end of the season. And of course, a huge number of points in between. Obviously, to interpret this, we'd really like to know what the yield data would be for this data set. Given there is this variability, ultimately, if water is a limited resource, and it certainly is in this part of Spain, you need to be able to apply this water most efficiently to maximise productivity. And that's very much what my research interests are. So today I wanted to sort of cover, I guess, 3 case studies. One, a technique known as partial root zone drying, which I suspect some of you may have heard me talk about before. More recently, we've been doing some work on what we would call sustained deficit irrigation. And lastly, I want to consider some of our more recent work on rain-fed wheat in the UK and how root angle may be important in this particular system. I guess the overall theme then is manipulating irrigation to try and understand its impact on root shoot signalling. So in terms of how the roots might communicate with the shoots, well, clearly roots are of primary importance in taking up water and distributing that to the shoot. And certainly I would not want to argue against the idea of hydraulic signalling, that the amount of water taken up by the roots and particularly the tension in the xylem will have an impact under certain circumstances on the growth and the physiology of the shoot. So clearly in this picture of here with wilting tobacco, it's hard to believe that hydraulic signals are not responsible for the decrease in leaf gas exchange and the decrease in growth of that particular crop. But I would argue that that's a very artificial situation for two reasons. One, that's the result of a PhD student not watering their experiment. Okay? The other point, perhaps a little more seriously, the root systems of those plants are constrained. So consequently, they're absolutely dependent on the regular supply of water to ensure that the plants are able to grow and photosynthesize optimally. That is rather different from the field situation where the roots are able to grow through the soil and potentially get access to water quite deep within the soil profile to sustain turgor and to sustain leaf water potential. But even in situations where the crop can sustain turgor and leaf water potential, stomatal conductance and growth can still be limited. And the argument is that chemical signals, in particular plant hormones moving from the roots to the shoots, can actually regulate some of these processes within the leaf. Now, this is not a new concept. This idea of chemical root-shoot signalling has been around since the 1980s. And indeed, some of the work from CSIRO Plant Industry in Canberra was some of the pioneering work within this area. There was another very influential experiment done in the 1980s, and for a rather different reason, when I first went back to Lancaster to work in 2001, I repeated this experiment with one of my colleagues, Wagdi Sobi. And the experiment was a situation of partial root zone drying. So we grew our tomato plants, in this case, in specialised split pots. You can see that up the top there. And so what we did is we set up two different irrigation treatments. In some plants, we watered the entire root system. In the other treatment, we only watered part of the root system, and we allowed the other part of the root system to dry the soil. We made a series of measurements, including at the top there, measurements of leaf water potential with a pressure chamber. And you can see that there is no difference in leaf water potential between these two sets of plants. Now, if any change in physiology was governed by water potential, you might expect there to be such a change in leaf water potential. We can see in the second panel, though, that the plants that were exposed to partial root zone drying closed their stomata. And this occurred in the absence of any change in leaf water potential. So I would argue that it's quite difficult to make the argument that it's changes in leaf water relations that are causing this stomatal response. Instead, I would make the argument that it's the stomatal closure of the plants grown with partial root zone drying that has actually allowed those plants exposed to dry soil to maintain their leaf water potential. And indeed, this was I guess a classic experiment when it was first performed in the 1980s. In conjunction with a number of other experiments, it sort of led rise to the idea that soil drying would result in changes in soil properties and which would stimulate the production of certain chemical signals such as some of these plant hormones here. This sort of schematic is something that I've interpreted from the literature. I wouldn't argue that every line is strictly true. I still think there's a lot of work that we need to do. The root-to-shoot signalling literature has sort of taken two main areas of focus. One is to try and understand what the impacts of these chemical signals may be on shoot physiology. The other aspect is to try and understand what is actually happening in terms of soil physics that is giving rise to these changes in chemical signalling. This black box has to some extent been relatively ignored because of the difficulties of actually making measurements on the roots. And I'm sure there's a number of people in the audience who would agree with that. I think what really interests me is how we might be able to apply knowledge such as this to try and improve agricultural productivity. And indeed, one of the earliest examples of this, using information about root-to-shoot chemical signalling to inform irrigation practice, was developed by Brian Lowies at CSIRO in Adelaide. And he basically extrapolated that laboratory split root experiment into the field. So he was working with grapevine, and he set up two different treatments. Full irrigation, where the entire root zone was irrigated, and then a partial root zone drying treatment, where there were two drip lines either side of the row of vines. One was watered, and the other one was allowed to dry the soil. And then after a period of time, the wet and the dry sides were swapped over. Now, clearly there's a lot of data there. I want you to draw your attention to the two red arrows. So we can see that the partial root zone drying treatment decreased irrigation by about 50%. There was no actual change in the fruit weight between the two different treatments. And this was a really positive result. It suggested that using this technique, you could potentially decrease irrigation and have no effect on fruit yield. And indeed, there were some very positive effects in terms of fruit quality. Now, I always show this slide to my undergraduate students, and I challenge them to say, well, what's wrong with this experiment? And those of you who are working in this area would realise that I've varied two things within this, or the two things have been varied within this experiment. Partial root zone drying has not only varied the placement of irrigation, but it's also varied the volume. So there is potentially some confounding there. This, of course, was recognised, and since this original pioneering experiment, there's been a large number of experiments that have compared the impacts of partial root zone drying and conventional deficit irrigation at the same irrigation volume. And I've summarised some of this literature in this graph here. So I would stress again, in comparing these two different treatments, both sets of plants received the same volume of irrigation water. Now, if yield is only determined by irrigation volume, you would expect there to be no difference in yield between these two treatments. And if I plot this yield ratio of partial root zone drying compared to deficit irrigation, you would expect all these studies, each bar represents a separate study in the literature, to fall on that line of 1. Now, clearly, there are some cases, individual studies, where partial root zone drying and deficit irrigation, when they apply the same amount of water, produce the same yield. But if we look above that line of 1, we can see many examples where partial root zone drying has out-yielded deficit irrigated plants. Now, that's a really interesting question. Now, why might this be the case? And I guess that's what I was particularly interested in trying to answer when I went back to Lancaster and got initially a postdoc working on partial root zone drying in Lancaster. And what I was very interested in trying to understand is whether this soil moisture heterogeneity generated by partial root zone drying would actually affect both root ABA accumulation and its long-distance signalling. So to try and understand how an ABA signal might be generated from the roots, I developed a rather strange-looking plant here. This, I believe, was the world's first ever two-legged sunflower plant. Now, the reason I created such a plant using a grafting technique was that I wanted to measure the ABA coming from each part of the root system. And so by inserting a signal shoot into next to these two sort of hypocotyls, I could actually then remove the shoot and measure the ABA coming out of the dry side of the root system, and also measure ABA coming out of the wet side of the root system. Also, I was able to put sap flow gauges on the two different parts of the root system, and therefore measure the water uptake from those two different parts of the root system. So I was able to collect a lot of data using such a system. And I've summarised some of it here. So here I plotted the xylem ABA concentration in the leaves as a function of either the leaf water potential on the top or the soil water content. And you can see that the two different irrigation techniques, deficit irrigation and partial root zone drying, show different responses. And what I particularly draw your attention to is this response to partial root zone drying. As the soil starts to dry, you get a stimulation of ABA, as the theory would predict. But later, if you continue to dry this part of the root system, the ABA concentration actually decreases. And indeed, this seems to make some sense. If you think about if you're trying to generate a chemical signal in those roots, it actually needs to be transported to the shoots to have its effect. And so, based on the measurements of sap flow, I sort of came up with a very simplistic model to show that my ABA concentration in my partial root zone drying plants was equal to the concentration from the wet side of the root system multiplied by the fraction of sap flow from that root system, and there's the corresponding term for the dry root system. And that model better explained the ABA concentration in the xylem of the leaf than simply assuming that ABA concentration was related to the whole pot soil water content. More recently, one of my colleagues has sort of done some measurements looking at the ability of this model to actually explain variation in xylem ABA concentration at different stages of the wetting and drying cycle. So you can see here with the plants exposed to partial root zone drying, if you look at the top panel, you can see that the soil water content was maintained in the wet side, but very rapidly in the dry side, the soil water content decreased. You can see the corresponding levels of sap flow through the two different parts of the root system. And on the basal panel, we can see here variation in leaf water potential and ABA concentration in the leaf. In each case, sort of the 3 times at which xylem ABA concentration was measured, it seems as though this sort of model, looking at the fractional contribution of different parts of the root system to ABA, better explained the ABA concentration in the leaf than either relying on leaf water potential or bulk soil water content alone. What's also interesting is where this ABA is actually generated within the root system. And a sort of common question I've been posed is, well, is this vertical split of the root system actually necessary to induce a situation of partial root zone drying? And so one of my staff members, Jaime Portales, has recently done quite an interesting experiment where he's tried to separate the effects of where you dry the soil on the actual process of partial root zone drying. So we have the sort of typical horizontal partial root zone drying, and then actually drying the upper layer of the soil and keeping the bottom part of the soil wet, which is indeed more similar to what happens in commercial practice if you get upper soil drying. What's particularly interesting about his results is that you can see that it's only with the sort of lateral partial root zone drying that you get significant ABA accumulation in the roots. And we suspect this is due to differences in the actual roots that we are sampling in terms of their morphological age. And I think this is potentially quite important because it suggests that depending on where you are actually drying the soil and which roots are actually exposed to drying soil will affect the ability of any partial root zone drying treatment to actually result in an ABA signal. So these are the local processes occurring in the roots. Did we actually get an ABA signal? And disappointingly, in this particular experiment done with potato, independently of how the soil drying was imposed, whether it was in soil columns or split pots, vertical or horizontal partial root zone drying, we basically got the same ABA concentration in the xylem sap. And certainly this particular experiment is somewhat different from a lot of the partial root zone drying literature where there is significant xylem ABA accumulation as a result of partial root zone drying. If we go back to look at, in this experiment, where the plants are actually taking up water from, we can see quite clearly that in this particular experiment, there is very limited water uptake from those roots in drying soil. Again, sort of confirming that this model is appropriate to explain the xylem ABA concentration occurring in the shoots. And so for me, the really interesting question is, under what circumstances can you maintain water uptake from the dry side of the root system but still generate ABA at the same time? In this particular experiment, we almost got a perfect match of ABA generation after the water uptake from those roots in drying soil had basically declined to nearly zero. And so I think this is a real challenge for the future, to try and understand this relationship between water uptake versus ABA accumulation and transport. That's not to say that the roots are unimportant. And this is one particular experiment where we deliberately manipulated the amount of roots exposed to drying soil by planting barley plants with seminal roots distributed between two compartments. You can see that that's the sort of schematic of how we started the experiment. Initially, the plants were well watered, and then we exposed them to soil drying for about a week. And then at the end of the experiment, looked at how much root material was in the soil that had been allowed to dry, how many roots were in the soil that was allowed to dry. What's particularly interesting, and perhaps not that unexpected, is if you expose more roots to drying soil, you decrease leaf growth more, you increase the ABA accumulation within the shoot. All that occurred with actual no change in leaf water potential between the three different treatments exposed to partial root zone drying. Sort of one last example showing the importance of the root system in affecting responses to water-saving irrigation techniques such as partial root zone drying is some work we've done recently with potato. So we've used two different kinds of potato, Maris piper, which, as anyone who's been to the UK, is the #1 chipping potato in the UK. Almost certainly if you've bought fish and chips in the UK, you've eaten this potato variety, Maris piper. And we've used something that was sold to us as a drought-tolerant potato variety from Egypt known as Horizon. And we can see that we've got very big differences in shoot vigour within these two different potato varieties. So Maris piper basically has very limited root system at the base of the soil profile. In contrast, our Horizon cultivar has a significant root mass at the base of the soil profile. Now, when we impose partial root zone drying on these two different cultivars, we can see that the Maris piper shows a decrease in tuber yield and a decrease in horn biomass, so the shoot biomass, with these partial root zone drying treatments. In contrast, the Horizon variety showed no real difference in yield as a response to partial root zone drying. One of the important issues within this partial root zone drying arrangement is to ensure that the wet and the dry sides are frequently alternated. And indeed, here's just to show the importance of this alternation, here's another meta-analysis where I've plotted the yield of the alternate partial root zone drying, so you swap the wet and the dry sides over, compared to those that were exposed to fixed partial root zone drying. And you can see in all the cases in the literature, alternating the wet and the dry parts of the root system seems to be particularly important in terms of maintaining yield. And there could be a number of reasons for this. One, maybe changes in xylem ABA concentration and other signalling from the roots to the shoots, but there's perhaps a more obvious explanation, and that probably doesn't need much in the way of explanation. So this alternate wetting and drying of the root system massively stimulates root growth. Now, this is from an experiment we did in pots, so it was under basically optimal irrigation, and so in that case, this extra root material would not potentially be advantageous in terms of requiring water, acquiring nutrients. This response also seems to be prevalent in the field as a response to partial root zone drying. And I suspect this is one of the major reasons why partial root zone drying manages to maintain yield with less water being applied, because of its ability to stimulate root growth, to allow those roots to access water deeper in the soil profile and maintain plant growth. So I want to turn now to, I guess, a different irrigation technique, sustained deficit irrigation. And this is a result of a sort of horticultural project where one of our local nurseries said to us, okay, we're interested in potentially applying less water. What's the best way to do it? Should we be stressing our plants for a period of several days and then re-watering them? Or should we be giving them a little amount of water very frequently? And I guess those that watch their weight may think, well, what's the best way? Should I be dieting? Should I go feast and famine or little and often? And I think this question for plant growth has not, at the moment, been satisfactorily addressed. So we worked with this ornamental plant pelargonium, not something you'd normally think of working with, because this is a project that was part sponsored by this nursery. We set up two different treatments. I guess an acute soil water deficit where basically water was withheld from the pots. And then a chronic soil water deficit where the plants were given small amounts of water very frequently. So what impacts did this have on the soil moisture content in these pots? So we can see with our chronic water deficit, it was applied for a much longer period of time, but by the end of the experiment, there was basically a sort of stable soil water content in those pots. Contrast that with our acute water deficit, where we can see that soil moisture rapidly declined in those pots compared to our well-watered plants. So that was the imposition of soil moisture content. What impact, if any, did this have on the leaf water potential of the plants? And what was quite striking was that we got a very different response of leaf water potential to soil water content depending on how we applied the treatments. So if we simply withheld water, this is sort of this classic response of leaf water potential declining as the soil dried. And indeed, that's some data to sort of back up those initial images I showed you of those wilting tobacco plants. With our chronic water deficit, where we're applying a little bit of water fairly frequently to the pots, we can see a very weak or indeed a positive relationship between soil drying and leaf water potential. So clearly, these plants that are regularly re-watered once or twice a day show a difference in their signalling system compared to those from those plants from which water is withheld. When we looked at the stomatal conductance of the two sets of plants, we saw a fairly similar relationship. If anything, those plants that were exposed to the acute water deficit, so simply withholding water, showed a much greater decrease in stomatal conductance for the same level of soil drying. If we looked at trying to explain these changes in stomatal conductance in terms of leaf water potential, again, depending on how we watered or not watered the plants, we got a very different response of stomatal conductance to leaf water potential. So if we withheld water from the soil, stomata closed as the leaf water potential declined. Within our chronic water deficit or our sustained deficit irrigation treatment, we got a very different relationships. So stomatal conductance was actually reasonably high as a function of decreased leaf water potential. What was interesting in this experiment was that we got a very uniform relationship between the stomatal conductance and the xylem ABA concentration measured in the leaf. And we've gone on to show that these sort of concentrations of ABA that we measured, if you supply them to a well-watered leaf, that is actually sufficient to close the stomata. But for me, the really interesting graph is this one here. we think about xylem ABA concentration being a function of soil water content. But depending on how we actually imposed our different irrigation treatments, we got a very similar looking relationship, but we got a massively attenuated ABA response to this sustained deficit irrigation treatment. And we're currently trying to understand the basis of this response. So in terms of thinking about our control systems, in both these two different treatments, we got a decrease in soil water content or gravimetric water content, a decrease in stomatal conductance, an increase in xylem ABA concentration. But the response of leaf water potential was very different in these two treatments. In trying to understand why we've got these different responses, I think we need to look at the soil moisture to try and understand what's actually going on in these treatments. And certainly, you know, you could look at the sort of colour of blue on these graphs to try and understand where the soil moisture is within these pots. So under well water conditions, uniform soil moisture. In frequent deficit irrigation or withholding water, you get a uniform depletion of soil moisture within the soil profile. But with this regular re-watering of the pots, with our sustained frequent deficit irrigation, we can see that we are getting a high water content in the upper part of the soil profile and yet depletion of soil moisture in the lower part of the soil profile. So if you like, a reverse of the vertical partial root zone drying treatments that I introduced to you earlier. It seems as though the root ABA accumulation will be affected by the gradient of soil moisture that is actually imposed. I gave some evidence for that in terms of the potato experiment with different root ABA accumulation according to where the roots are actually sampled. This graph here shows a rather interesting response, again, to different soil moisture gradients. So we can see that if we maintain a lot of water on the base of the pot, we get very limited ABA accumulation as a function of soil drying. If we simply withhold water, again, we get high root ABA accumulation for the same level of soil moisture. And I think this response here may be involved in the response of sustained deficit irrigation that we saw earlier. And so our student Richard, who's currently working on this project, has been doing some work with tomato grown in soil columns, and he aims to measure the root ABA accumulation and also the soil water uptake from different parts of the root zone to try and explain this different response to stomatal conductance to these two different treatments. And indeed, we can see here for the plants that were exposed to the sustained deficit irrigation, we get, if you like, a sort of inverse from soil water profile. So we have high soil moisture in the upper part of the profile and very low soil moisture at depth within the soil profile. And you only get the root ABA accumulation in the lower part of the soil profile, coincident with low soil moisture. He's currently working his way through the soil water uptake calculations, but I suspect that most of that ABA that is accumulated within the lower part of the soil profile is not actually being transported to the root, to the shoots. And that explains why there is this attenuation of xylem ABA concentration with this sustained deficit irrigation treatment. So, just in the last sort of 5 minutes or so, I just want to touch base with what we've been doing with wheat. So let's move away from sort of irrigated systems to rain-fed systems. And if you've ever been to the UK, even in the summer, you will know that often UK rain-fed systems are very well rain-fed. Having said that, in the south of the country, if you irrigate your crop and it's grown on some of the lighter soils, it's possible to increase yield by 1 to 3 tonnes per hectare. And so this is an experiment we did with Rothamsted research in the southeast of the country. Those of you who know a little bit about agricultural research history will realise that Rothamsted research is the oldest agricultural research station in the world, and they have a sort of continuous wheat experiment that's been going since the 1850s. This is our irrigation trial, and it's clear to see that if you irrigate the crop, you can maintain green canopy, and as a result of that, improve yield. Our PhD student, Laura, is very interested in the role of root angle in UK rain-fed systems as a result of some of the work done here in Australia. So certainly there's a lot of evidence, and I think Graham here in the audience was involved in some of this work, showing that having a relatively restricted root angle allowed those roots to access soil moisture deep in the soil profile. And this can have a substantial impact in terms of maintaining the yield of rain-fed wheat. In the UK, where it's a rather different soil moisture pattern, where you can frequently get rainfall throughout the growing season, is this the kind of rooting pattern that we'd actually want? And I think there's some questions as to whether this is going to be advantageous or not. What Laura has done is used, I would say, a rather low throughput phenotyping technique that has allowed her to understand the root angle of these seminal roots of wheat. So she's used an adaptation of the sort of basket pot method and actually determined through which of these pores in the net pot do the roots actually emerge from. And she's done this experiment with soil that is wet and also mildly dry. And what's interesting is that for some genotypes of wheat, there's absolutely no difference in the angle of the roots depending on whether the soil is wet or dry. They emerge at the same angles. Contrast that with this one variety, Robigus, which does something rather unusual in the sense that the roots become more shallow when the soil is really wet. Now, what the mechanism of this response is, we're not completely certain of. But this does seem to have some kind of impact in terms of its root length distribution within the field. So if we look at sort of the root length as you go down the soil profile, Robigus has much fewer roots, puts much fewer of its roots deeper within the soil profile under our soil conditions. Of course, knowing where the roots are is one part of the problem. You actually need to try and understand what their physiology and what their function is. And so we've been making some measurements looking at the water uptake of these wheat roots. And what's interesting again with Robigus is that you get much greater changes in soil water content in the upper part of the soil profile compared to some other varieties, and then very low in the soil profile where Robigus does not put much roots at all, seems to result in very limited water uptake. So having sort of made these measurements, ultimately the question might be, well, does this have any impact on crop yield? And I think that's a very difficult question to answer, given that these different wheat varieties that Laura has been working with are in very different backgrounds and indeed have very different susceptibilities to other pest and pathogen problems. And it's interesting that I think that Robigus is relatively stable in terms of its yield on different sites compared to some of the other varieties that seem to be more affected by different soil conditions. So, to sort of conclude, starting with the roots, given that was the title that I gave to the seminar, certainly root ABA concentration is dependent on local soil moisture. It also appears that the root water potential is important in modulating that response. But also, I would make the argument that not all roots are equal in terms of their ABA accumulation, due to changes in where they are within the soil profile. I realise this has been a very ABA-centric seminar, and there's a number of reasons for that. One is it's relatively easy to measure. two, I believe it's very important in controlling leaf gas exchange and water use efficiency. It will also have an impact on root growth. At the moment in our lab, we're very interested in also looking at ACC signalling or ethylene signalling within the plant. And I think this is potentially quite important given that ethylene has been known in many circumstances to be a growth inhibitor. Hopefully I've convinced you of the case of chemical signalling being important in plants. And in terms of how we might apply that, I think there's been a lot of progress with this technique of partial root zone drying. It is still primarily an experimental technique that agronomists and those in research institutes are applying. There are some examples, particularly within China, where it's applied to maize crops over a much wider acreage. But it still remains, I would say, fairly experimental. And given the complexities of actually understanding under what conditions would you get ABA moving from the roots to the shoots, it's not surprising that it is somewhat empirically applied and the results can be a little unpredictable. I would argue that sustained deficit irrigation is potentially much easier for the practitioner to apply, but clearly, and this could potentially be automated, but clearly the irrigation frequency has a large impact on the water use efficiency and the signalling of those crops. Lastly, under rain-fed systems, certainly the work from here suggests that changes in root angle can have a big impact in terms of enhancing water capture. Whether we'd be looking for a different idiotype of root angle in UK conditions is something I think we need to try and understand. And it just remains for me to thank those people whose work I've talked about today, particularly Richard, who's done the work on sustained deficit irrigation, Laura, who's done the work on root angle, and Jaime and Juan, who've been doing a lot of work on partial root zone drying, and of course the funders. So thanks for your attention.